Good morning. Today we are going to study the structure of nucleotide. Okay. Today we are going to study the structure of nucleotide molecule. Okay. Actually, there are two types of nucleic acids, that is the DNA and RNA. DNA is the deoxyribose nucleic acid and RNA is the ribose nucleic acid, and these nucleic acids are polynucleotide molecules. Okay. Polynucleotide means consist of many nucleotides. So today we will discuss the structure of one nucleotide molecule, because the DNA and RNA are the polynucleotide, means they consist of many nucleotides. In most of the organisms, DNA acts as the genetic material, while in some viruses, RNA also acts as the genetic material. Okay. Now, each nucleotide molecule consists of three components. So, what are the three components of each nucleotide? So, each nucleotide consists of bases. That is the nitrogenous bases. Okay. So each nucleotide consists of nitrogenous bases. Nitrogenous bases are two types. That is the purines and the pyrimidines. Purines are again two types, that is the adenine and guanine. Pyrimidines are three types, that is the thymine, cytosine and uracil. So these are the pyrimidine bases. So in each nucleotide there are the three components, that is the nitrogenous bases. Any one nitrogenous base found in each nucleotide. Nitrogenous bases are two types, that is the purines and pyrimidines. Purines are two types, that is adenine and guanine. Likewise, pyrimidines are three types, thymine, cytosine and uracil. So these are the nitrogenous bases. So one of the nitrogenous bases found in each nucleotide. Now we will talk about what is the difference between these purines and pyrimidines bases. So purines are, first we will talk about the purines are. So purines are purines are nine-membered double ringed structure, or you can say that the dicyclic structure. So which type of structure? So double ringed structure. This is the one ring, and now there is the another ring. So this is the nine-membered double ringed structure, or you can say that the dicyclic structure. Okay. So purines, we are talking about the purines. So purines are nine-membered structure, or you can say that the double ringed structure or dicyclic structure. Nine-membered means there is the nine-membered 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6, 7, 8, 9. Now this is the ninth position. At ninth position, there is the NH group. That is very important group, this NH group at the ninth position. This is the NH group that take part in the formation of glycosidic bond with the sugar. So this purine bases attached with the sugar at ninth position. At ninth. So there is the NH group at the ninth position, and this NH group take part in the formation of glycosidic bond with the sugar. Okay. So this is the purine bases, adenine and guanine. Now we will talk about the thymine, cytosine and uracil. They are six-membered. They are six-membered single ringed structure, or you can say that monocyclic structure, single ringed or monocyclic structure. What is the structure of this pyrimidine basis? There is a single ringed structure. Suppose this is the single ring. This is 1, 2, 3, 4, 5, 6. Six-membered structure, monocyclic structure. Now suppose this is the first position. Suppose this is the first position. At the first position, there is also NH group that is present, and this NH group now take part in the formation of glycosidic bond with the sugar. Means the pyrimidine bases are six-membered, or you can say that the monocyclic, and the nitrogen that present at the first position take part in the formation of glycosidic bond with the sugar. Means the sugar attached with the pyrimidine bases with the nitrogen present at first position, and the purines are attached with the sugar, that is the nitrogen present at the ninth position. So this is the difference between the purines and pyrimidines. Okay. Now we will talk the second component. This is the nitrogenous bases we discussed here. Now we will talk the second component is the pentose sugar. Now we will talk about the second component, that is the pentose sugar. So there are two types of this pentose sugar. One is the deoxyribose sugar, and another one is the ribose sugar. Another one is the ribose sugar. So these are the two types of pentose sugar. Pentose sugar means 5 carbon sugar. So there are two types, deoxyribose and the ribose. Now we will discuss or study the structure of this deoxyribose and the ribose sugar. Okay. Now you can see here. Now suppose this is the structure of a sugar. Now the carbon, there is the first carbon, second carbon, third carbon, fourth carbon, and fifth. This is the 5 carbon structure. These are the, so now you can see this is the 5 carbon. Now there is the oxygen. Now this is the OH group. There is the hydrogen. There is also H, OH. Suppose there is also the H, OH. There is the H. And now here I will write CH2OH. Okay, at the 5th position. So this is the first carbon. This one is the second carbon, this one is the third carbon, this is the 5th, and this is the 4th, and this one is the 5th carbon. So this is the pentose sugar, 5 carbon structure. Now you can see this is the structure. Now another structure. Now this is the actually ribose sugar. This one is the ribose sugar. Now we will talk the structure of deoxyribose. Now what is the difference between ribose and deoxyribose? We will discuss. Now see. There is the carbon, first, second, third, fourth, and the 5th carbon. Okay. Now there is also C, H, 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 OH, H. Now here I am writing the CH2OH. Okay. So this is the first carbon, second carbon, third carbon, fourth carbon, and the 5th carbon. Now you see the difference between this ribose and deoxyribose. Okay. This is ribose sugar and this one is the deoxyribose sugar. Now you see this position. This is the position now only you see. At the second carbon, now you see in the ribose sugar, there is the hydroxyl group is present, while in the deoxyribose, there is no hydroxyl group. So this is the difference between the ribose and deoxyribose sugar. So at the second carbon, there is the difference at the second position. At the second carbon, there is the hydroxyl group present, while you can see that there is no hydroxyl group. There is only hydrogen, I have written here. Only hydrogen. There is no hydroxyl group. So this is difference between the ribose and deoxyribose. Then what will be the molecular formula? Molecular formula for ribose and deoxyribose. So what will be the molecular formula then? For the ribose, there is the C5, H10 and O5, there is the, and now for the deoxyribose, the formula will be C5, H10 and O4. So this is the difference between the ribose and deoxyribose. So in ribose, there is the, you can see that this is the formula C5H10O5 molecular formula, while in the deoxyribose there is the O4. So deoxy means one oxygen less. Deoxy means one oxygen less. So difference between this ribose and deoxyribose is at the second carbon position, that you can see that at the second position there is the hydroxyl group, but where there is no hydroxyl group, and this group, this position is very important group that we will discuss in the next video. Okay.